to introduce yourself?
Sure. My name is Fridman. I'm from Johns Hopkins University.
Okay, and very briefly, what is your research about, or what are you presenting here?
My research is about developing a device that can deliver ionic direct currents safely to the tissue, which is something that has not been normally practiced in this field.
Okay, and what are some challenges? Why hasn't it been done before, and what do you hope to accomplish with this?
So the reason why it hasn't been done before is because when you deliver direct current to a metal electrode implanted in the tissue, you get bad responses. You get electrochemistry, electrolysis, corrosion, and pH changes, all kinds of bad stuff. And so what we're doing is we are taking metal electrodes, putting them inside of a device, providing what is considered to be standard stimulation pulses back and forth, so there is no electrochemistry happening. But as that happens, we also have the machinery, or we have the microfluidic machinery in form of an H-bridge or a bridge rectifier in the device, such that the output of the device is direct ionic current. And so there is no electrochemistry being done inside of the device, and the output is ions moving through. So there's presumably no effect, no negative effect on the tissue as well.
Interesting. Okay, so ions are basically what's transmitting the charge instead of like voltage across metal, something like this. So yeah, what do you hope to achieve with this, or what are some next steps with this technology?
Sure. So in this particular poster, we're looking at the ability of ionic direct current to preferentially block pain fiber activity. The small diameter pain fibers that normally carry pain from the periphery to the brain, or to the spinal cord. What we're seeing is an ability to block those pain fibers or pain axons at a lower amplitude than it takes to block the sensory or the motor fibers.
So maybe habituation or something in this case would not be such a big issue because the stimulation would be lower, something along these lines?
I don't know about habituation. We're not to the point where we can study long-term effects. The experiments that we have done are acute, and these experiments last over 4 hours. We are able to deliver and reliably suppress activity of the pain fibers over that duration, while seeing the propagation of the sensory information to the spinal cord. This poster also explains the potential mechanism behind why we think that this works.
Okay, and what is that?
The reason we believe is that direct current. So the mantra in this field about the thresholds for depolarization or affecting neurons with electrical stimulation is that the large fibers get affected at a lower amplitude than small fibers. That, however, is true for short duration pulses. What we're finding instead is that as the pulse gets longer, that difference between the threshold disappears. And so when we track down why, then do we see this difference in being able to block small fibers versus large fibers? What we're finding is the likely effect here is due to the difference in the voltage-gated sodium channels that are expressed in small fibers versus the large fibers. And it turns out that the small fibers can be blocked, or can block the sodium channels of those fibers at a lower potential than those for a large caliber neuron. And we're showing that in a computational model.
Okay, interesting. So basically, you're able to selectively, stop some of these channels. And what does that mean? Like, what's the difference? Like, what are small fibers usually associated with, and what are large fibers usually associated with?
Small fibers typically are associated with conducting pain or nauseous information. And large fibers conduct typically muscle innervation and sensory information from your skin.
Okay, very cool. Are there any parting thoughts?
No.
What is maybe some next steps? What do you? I mean, not to reveal any secret information, but what's kind of the next step for you guys?
Sure. No, next steps are pretty clear. We need to understand. So this model was made in almost an idealized kind of a case where we try to isolate the response of the particular channels. But of course, there are other differences between these types of neurons, so we need to investigate what those differences are, and do we still maintain this effect. We also want to look at the pharmacology to be able to drive this to a conclusion in terms of the mechanism. And of course, we want to conduct studies in a behavioral animal once we complete the development of our microfluidic device.